When a drug enters the human body, its journey does not end at the point of administration. Instead, a complex process begins, known as drug distribution. This process determines how, where, and when the drug molecules reach different tissues and organs after being absorbed into systemic circulation. Imagine the bloodstream as a vast river system. Once a drug is absorbed, whether orally, intravenously, or by another route, it flows through this river, moving from regions of high concentration to those of low concentration. The distribution is not random. It is directed by a series of factors, such as blood flow, tissue permeability, binding to plasma proteins, and the chemical nature of the drug itself. The first organs to receive the drug are typically those with the most substantial blood supply: the heart, liver, kidneys, and brain. These organs act like the busiest ports along the river system, receiving preferential deliveries. However, reaching some destinations requires overcoming additional barriers. For the brain, this means passing through the blood-brain barrier, a stringent checkpoint that only allows certain drugs, those that are lipophilic and small in size, to cross easily. Another important concept is plasma protein binding. Many drugs bind to proteins such as albumin in the blood, acting as if they have boarded a ferry rather than sailing freely down the river. Only the free, unbound, drug can leave the bloodstream and enter tissues to exert its therapeutic effect. If a large proportion of the drug is bound, its distribution is limited, and only a small fraction is available for action or elimination. Different tissues have varying affinities for drugs. Fatty tissues, for instance, can act as reservoirs, soaking up lipophilic drugs and slowly releasing them over time. Bones and teeth can sequester drugs like tetracyclines, leading to prolonged storage and sometimes unintended effects. As the drug continues its voyage, several factors can modify its distribution. Diseases affecting organs, age, body composition, or interactions with other drugs can all influence where the drug goes and how long it stays. For example, liver disease may reduce plasma protein levels, increasing the free drug fraction and, consequently, both its effect and risk of toxicity. Distribution is not a static event, but a dynamic balance. Eventually, as plasma concentrations decrease, drugs may move from the tissues back into the bloodstream, continuing the cycle until they are finally metabolized or excreted from the body. Understanding the mechanics of drug distribution is essential for designing effective and safe dosing regimens. It ensures that the right amount of drug reaches the correct site of action, no more, no less. It also helps doctors anticipate potential side effects, drug interactions, or complications in special populations. Ultimately, drug distribution mechanics are about precision, delivering therapy where it is needed most, at the right time, and in the right amount.